In Africa, medical teams are scrambling to contain a deadly outbreak of Ebola, a highly contagious, often fatal virus with no known treatment.
విదేశాల నుంచి వచ్చి విమానాశ్రయాల నుంచి ప్రయాణికులు ప్రతి ఒక్కరికి వైద్య పరీక్షలు నిర్వహిస్తున్నారు.
For 40 years, the world appears to be on the brink of a global epidemic of a new flu virus.
Infectious diseases interact between organisms, their environment, and entire ecosystems can be affected. Anthropogenic factors that influence the presence of infectious disease include water management, how land is developed and utilized, and waste disposal. This can then lead to the development of a niche that a disease-carrying species can exploit. Evolution of the disease and its ability to spread to its new habitats is also tied to climate itself. The bubonic plague is a disease that is caused by a bacterium that is common in some rodents in urban environments. Within the rat community, certain populations could either be immune or succumb to the bacteria. In the Middle Ages, bad hygiene and poor sanitation led to the proliferation of these rats in cities. The lack of knowledge of transmission led to an epidemic. Research says the disease likely originated somewhere in South China. It spread as far as Europe via trade and military transports carrying infected rats and fleas. Conditions that exacerbated the plague include widespread famine, biological warfare, and climate change. Interactions between humans and rats under unsanitary conditions allowed the fleas to parasitize both organisms. As the spread of disease became more understood, measures such as improved hygiene, burning of bodies of the dead, and quarantine led to decline in infection. An ecological factor that led to the decline of the plague was the introduction of the black and brown rat via trading routes. These rats were larger, exploited their niche, and were able to outcompete plague-bearing communities for resources. The plague shares similarities with another infamous killer, malaria.
Public enemy number one, Anopheles, the malaria mosquito, wanted for willful spreading of disease and theft of working hours.
Malaria itself is caused by a protozoa whose incubation is inside of humans, then matures inside the Anopheles mosquito, which then transmits the parasite to other humans or mammals. Climate is a main control in the life cycle of the vector, and too much rain, humidity, or periods of time with high temperature prevent these mosquitoes from completing their life cycle. Ecosystems with heavy rainfall can create stagnant pools of water, making suitable breeding sites for the Anopheles mosquito, and where the eggs are deposited and the larvae can develop. These climatic constraints make it possible to identify ecosystems that are susceptible to malaria infection, but as climate changes, habitats are shifting. In some areas, controlling the Anopheles population has lowered the risk of transmission, where other areas, environmental measures, pesticide application, antibiotics, and experimental vaccines are utilized to limit fatalities associated with infection. Reducing standing water, filling in depressions, and improving drainage in and around communities is the most important. To kill larvae in standing water, Paris green, insecticides, and mineral oils are used, but pollution of the ecosystem is likely to result from these measures. There are also biological controls being employed with varying success to curb the population of the mosquitoes. The introduction of the natural predators, fish, and other insects, including nematodes, has proven effective. Today we have a good understanding of how diseases are spread in the United States, but other countries lag behind our medical science. Poor understanding of medicine in these regions is leading to a rise in antibiotic-resistant strains and unknown or incurable diseases. The unknowns of political instability, disasters, or the inability to maintain infrastructure are ever-present variables that could give rise to future infectious diseases.